Hello, my name is Clay Swackhammer, and I'm a postdoctoral researcher here at Purdue University in the Department of Food Science, um, funded by a USDA NIFA postdoctoral fellowship. And today I'd like to show a very quick demo for one specific AI-assisted task that I've been doing for microbiome data analysis. So starting out, we're in Chime. This is a bioinformatics program. And I only want to do this step to kind of show where I'm getting the data from, taking one of these output files from Chime. If you're not a user of this program, just hang tight for a moment. Basically, what does this data represent? Well, it represents the estimated abundance of all of these different microbial taxa. That's basically like species, like type of microbe, within these different 27 biological samples. So what I'm going to do is go to taxonomic level 6. That's basically the genus level, and then download a spreadsheet, a CSV, called genus level abundance. So this is the raw data. This is how I got it into this format. If you were going to try and repeat this, maybe that'd be helpful. So we have like an index is some kind of sample ID, and then in each column we have this long name, kingdom, phylum, class, order, family, genus, for those different taxa. In each cell we have the estimated abundance or count of that sequence corresponding to that taxa. And then over here we have some metadata. So let's just get rid of that. And let's go over here and start a new project. Let's just call this AI assisted microbiome data analysis. And see if we can add this file. So, genus level abundance, that's the file we were just looking at. All right, let's see what we can do with this data. So let's create a new chat. Hey chat. Oh, boy, it already knows. Let's do it. Well, I don't know. Well, if you insist. Okay, you've uploaded a genus level abundance data set. Want me to take a look and summarize it? Maybe check for missing values, basic stats. Great plans, great ideas, but what I would like to do is just summarize it. Let's just summarize it for me, please. I have no idea what it's going to do here. This is kind of a, like, how would you summarize that spreadsheet? Just highly unclear. But all right, let's see. Dataset summary. There are 27 samples. Genus level abundance data is present for 100 plus taxa. The dataset includes metadata columns, time, fiber, and replicate. Fiber has 9 unique values, suggesting multiple experimental conditions. Time has a mean value of 21.3, let's be some are 0 and some are 24. Replicate has a mean of 2, indicating multiple replicates. I mean, it's just like charmingly obtuse, but it's all correct as far as I can tell. So let's see. How about this? Can you help me by identifying the taxa that are known to be butyrate to produce the short chain fatty acid butyrate? Let's make this a little harder, and then create a new variable in the table which is defined as the sum of the counts of all the butyrate producers in that row. Okay, so this is something you might want to do in microbiome data analysis. I mean, these are the butyrate producing genera: Faecalibacterium, Rosaburia, Eubacterium, so on and so forth. So in that long list of taxa right here, how many do we have? Well, how many columns are in this thing? Quite a few. You can see these kind of long taxonomic names that we're scrolling through. My question is, can we just identify the ones that are known to produce butyrate? That's really all I'm interested in, I guess. That's a very beneficial short chain fatty acid, certainly something we might want to analyze. Okay, so let's see. I'll scan your data set for these taxa and add the new variable. I've added the new variable. That's awesome. Can you highlight the columns? Let's see, the columns of the butyrate producer in yellow and make it downloadable in Excel format. Let's just take a look at it, see if we can do this. We uploaded a CSV, so it'll be interesting to see if it can convert that to an Excel natively, Okay, I mean, Shouldn't be too hard, right? So let's see, updated, genus abundance. See if our butyrate producers are now in yellow. Aha, look at that. Yep, Blautia, Faecalibacterium, Clostridium, probably Rosaburia, Ruminococcus, Eubacterium, Lactobacillus, Coppercoccus. Yeah, I think these all so far, Doria, yeah, okay. These certainly pass the test so far. Now, I would definitely double check this. I would double check this, and let's also take a look at this new variable over here that we produced. Butyrate producers, sum of butyrate producers. So I did this earlier and checked just the sum across the yellow highlighted columns, and that was correct. But let's do something a little bit more just to see if we can get some more juice. Hey, can you please make a bar graph where, let's say for each fiber, where the height of the bar is the average of the variable sum of butyrate producers for the 3 replicates of that fiber. Let's see if we can make a bar graph to see which of our experimental treatments, which is the type of fiber that was in this experiment. Let's see if we can get a graph. Wow, okay, cool. Can you add an error bar for the standard deviation? Okay, not bad. Okay, let's take this to the next level. Can you repeat the entire analysis, but for acetate producing taxa, and remake the bar graph? Now this, I think, would take you an afternoon to do without AI, especially if you were gonna like literally look up every single one of those taxa, just on account of the fact that there's hundreds of them. How much can you trust it? I really don't know. I think that finding some kind of protocol to verify key parts of this will be crucial to whether or not this really succeeds as a research tool. Because for me, you wouldn't want to just go with this output in a publication. But then again, verifying every single part of it by hand doesn't really seem feasible either, because that would sort of defeat the purpose of the thing. So if anybody has any solution to that problem, keep thinking about it. In any case, I hope you have enjoyed a little bit of AI-assisted microbiome data analysis. We've been able to have ChatGPT do some work for us, identifying taxa in the literature, creating graphs, visualizing the counts of those taxa, and I hope it at least catalyzes some new ideas for you. Thank you very much for watching.